Hey, science fans! Today we're diving into the microscopic world of bacteria. Don't let their size fool you. These single-celled organisms are genetic marvels. Bacterial genetics is the study of their DNA, the blueprint that makes them tick. Understanding bacterial genetics is crucial for many reasons. It helps us understand how bacteria cause disease, how they develop resistance to antibiotics, and how we can develop new treatments. It's like having a secret decoder ring for the bacterial world. Just like us, bacteria have DNA that carries their genetic information. This DNA contains genes, which are like instructions for building different parts of the bacteria. These parts can include things like the cell wall, proteins, and toxins. Studying bacterial genetics helps us understand how these tiny organisms function. It gives us insights into their strengths and weaknesses, allowing us to develop strategies to combat harmful bacteria and harness the power of beneficial ones. One of the coolest things about bacteria is their ability to share genes. Imagine if you could borrow your friend's super speed gene. That's essentially what bacteria can do. This process is called horizontal gene transfer, and it's a major reason why bacteria evolve so quickly. There are three main ways bacteria share genes: transformation, transduction, and conjugation.
Transformation is like finding a USB drive on the ground and plugging it into your computer. Bacteria can absorb free-floating DNA from their environment. Transduction involves a little helper called a bacteriophage, which is a virus that infects bacteria. These phages can accidentally pick up bacterial DNA and transfer it to other bacteria they infect. It's like a genetic courier service. Finally, conjugation is like building a bridge between two bacteria. They connect through a structure called a pilus, and DNA is transferred directly. This process is particularly important for spreading antibiotic resistance genes.
Now let's talk about plasmids. These tiny loops of DNA are like bacterial accessories carrying extra genes that give bacteria special abilities. Think of them like power-ups in a video game.
Plasmids can carry genes for antibiotic resistance, making bacteria immune to the drugs we use to fight them. It's like having a shield that deflects antibiotic attacks. These plasmids can be easily shared between bacteria through conjugation, spreading antibiotic resistance like wildfire. This is why understanding plasmids is crucial in the fight against antibiotic resistance. Scientists are constantly studying plasmids to understand how they evolve and spread. This knowledge is essential for developing new strategies to combat antibiotic resistance and keep our bacterial foes at bay.
Mutations are like typos in the genetic code. They're changes to the DNA sequence that can happen randomly. Most mutations are harmless, but some can be beneficial, giving bacteria an advantage. Imagine a mutation that makes a bacterium slightly more resistant to an antibiotic. When that antibiotic is present, the resistant bacterium will survive and reproduce, while others die off. This is called natural selection. Over time, these beneficial mutations can accumulate, leading to the evolution of new bacterial strains with increased resistance. It's like bacteria are constantly adapting and evolving to overcome the challenges we throw at them. Understanding how mutations contribute to antibiotic resistance helps us develop strategies to slow down the process. It's a constant race against bacterial evolution, and we need to stay one step ahead. Bacteria are masters of teamwork, and their genes are no different.
Genes often work together in groups called operons to control important functions.
Think of an operon like a group chat where genes communicate and coordinate their activities.
One famous example is the lac operon, which controls how bacteria digest lactose, a type of sugar. When lactose is present, the lac operon turns on the genes needed to break it down.
When lactose is absent, the operon shuts down, conserving energy.
This coordinated gene expression allows bacteria to adapt to changing environments and efficiently utilize available resources.
It's like a well-oiled machine, with each gene playing its part to ensure the survival of the bacterial colony.
By understanding how bacterial genes work together, we can develop strategies to disrupt their carefully orchestrated processes.
This knowledge is crucial for developing new antibiotics and other treatments for bacterial infections. Antibiotics are wonder drugs that have revolutionized medicine. They've saved countless lives by effectively treating bacterial infections.
However, their overuse and misuse have led to a growing problem: antibiotic resistance.
Antibiotic resistance occurs when bacteria evolve ways to survive exposure to antibiotics that would normally kill them.
It's like bacteria are becoming immune to our weapons, making infections harder and sometimes impossible to treat.
This resistance can arise through mutations or by acquiring resistance genes from other bacteria.
The more we use antibiotics, the more we create selective pressure that favors the survival of resistant bacteria.
Antibiotic resistance is a major global health threat, and we need to act now to address it.
If we don't, we risk returning to a pre-antibiotic era where simple infections could once again become deadly.
Bacteria are crafty opponents, and they've developed various strategies to resist antibiotics. It's like they're constantly studying our playbook and coming up with new ways to outmaneuver us. One common mechanism is enzymatic degradation, where bacteria produce enzymes that break down antibiotics, rendering them ineffective. It's like having a tiny army of Pac-Mans gobbling up the antibiotic invaders. Another strategy is efflux pumps, which act like tiny pumps embedded in the bacterial cell membrane. These pumps expel antibiotics that enter the cell, preventing them from reaching their target. It's like having bouncers at a club, tossing out any unwanted antibiotic guests. Bacteria can also alter their target sites, the specific molecules that antibiotics bind to. By changing the shape of these sites, bacteria can make it difficult for antibiotics to latch on and do their job. It's like changing the locks on your doors so the antibiotic keys no longer fit.
Every time we use antibiotics, we're setting up a survival competition among bacteria. The bacteria with resistance genes have a significant advantage, like having a secret weapon in a game of tag.
As we continue to use antibiotics, we're essentially selecting for the survival and proliferation of resistant bacteria. This is why it's crucial to use antibiotics responsibly and only when absolutely necessary.
Overusing antibiotics, such as taking them for viral infections like the common cold, only fuels the development and spread of resistance. It's like trying to put out a fire with gasoline. It only makes the problem worse.
Remember, antibiotics are powerful tools that should be used judiciously. By using them appropriately, we can preserve their effectiveness for future generations.
Antibiotic resistance is not just a theoretical problem. It's a real and present danger affecting people worldwide. It's a global health crisis that requires urgent attention and action.
Infections caused by antibiotic-resistant bacteria are becoming increasingly common and difficult to treat. This leads to longer hospital stays, higher healthcare costs, and increased mortality rates.
In some cases, previously treatable infections are becoming untreatable, leading to tragic outcomes. It's a stark reminder of the importance of antibiotics and the dire consequences of losing their effectiveness.
The World Health Organization has identified antibiotic resistance as one of the biggest threats to global health, food security, and development. It's a complex problem that requires a multifaceted approach to solve.
The good news is that we're not powerless against antibiotic resistance. Scientists and healthcare professionals are working tirelessly to develop new strategies to combat this growing threat.
One important strategy is antibiotic stewardship, which involves using antibiotics responsibly and only when necessary. This includes prescribing the right antibiotic for the right duration and educating patients about the importance of proper antibiotic use.
Another key area of focus is developing new antibiotics that can overcome existing resistance mechanisms. Scientists are exploring new targets and approaches to develop drugs that bacteria haven't encountered before.
In addition to new antibiotics, researchers are investigating alternative therapies, such as phage therapy, which uses viruses that infect bacteria to treat infections. It's like enlisting the help of nature's tiny assassins to fight our battles. The fight against antibiotic resistance requires a collective effort. We all have a role to play in preserving the effectiveness of these life-saving drugs. Understanding the basics of bacterial genetics and antibiotic resistance is crucial for making informed decisions about our health and the health of our communities. By staying informed, practicing good hygiene, using antibiotics responsibly, and supporting research efforts, we can help turn the tide against antibiotic resistance and ensure a healthier future for all. Remember, knowledge is power. The more we understand about bacteria, the better equipped we are to combat the challenges they pose and harness their potential for good.